Good afternoon. I'd like to thank the Board of ESPID very much for this award, and I'm delighted to tell you about my work looking at Clostridium difficile and host response in children with diarrhea. I have no conflict of interest to declare. To give some background, Clostridium difficile, or C. diff, is a gram-positive bacillus. It's an anaerobe, and it forms spores. It exists in both toxin-producing and non-toxin-producing forms. It's a significant cause of nosocomial infection, particularly in the adult population, with a wide spectrum of disease ranging up to fulminant colitis and death. We do know that neonates and infants frequently carry C. diff as part of their intestinal flora, with colonization rates as high as 70% in the first year of life, falling to around 4%, which is similar to adult rates at 2 years of age. Little is known about the epidemiological significance of this colonization, or the mechanisms by which children avoid disease in spite of the presence of toxin. Multiple mechanisms have been proposed. It's also difficult to quantify what disease actually is, particularly in comparison to adults where there are clear scoring criteria. Practice also differs between units on whether or not to treat in the presence of C. difficile. Our research questions were as follows. What is the prevalence of C. diff in hospitalized diarrheal infants and children? What is the host response to the presence of C. difficile? Is there a microbiotal signature associated with the presence of C. diff or of co-infection with other pathogens? Our study population were children aged 0 to 16 who'd been admitted to hospital with diarrhea. They'd had stool samples submitted to the hospital lab for routine testing. We looked at rates of culture positivity for C. diff, the presence of toxin, and we compared ribotypes between age groups. We also looked at blood and stool inflammatory responses and presence of other enteric pathogens in a subset of samples. Similarly, we looked at microbiotal diversity in a subset of samples using 16S profiling. Of note, no participants were treated for C. difficile. We found that a quarter of children under the age of 2 were C. difficile positive, of whom just over 50% carried a toxigenic strain. We also noted that 11% of children over 2 were C. difficile positive, and almost 3/4 carried a toxigenic strain. We can see, looking at ribotypes, that in children under 2, there's a higher prevalence of 020 and 039, whereas in children over 2, there's a much more equal spread of different ribotypes, and these were in fact similar to those seen in adults in the same region. We noted that the CRP was higher in the presence of toxigenic C. difficile. We also noted a non-significant association between toxigenic C. diff carriage and a higher white cell count. Of note, however, these patients were also more likely to be neutropenic, suggesting a hematological malignancy. In the subset of samples tested for alternative pathogens, we noted 27 of these samples were positive for an alternative enteric pathogen, of which 21 were positive for norovirus, which appeared to occur quite frequently in the presence of C. difficile in 9 of these 21 samples. We noted no pro-inflammatory stool cytokine response in the presence of C. difficile. We noted an increased Shannon diversity in terms of bacterial species in abundance in the presence of C. difficile, with an altered milieu in the presence of toxigenic C. difficile, namely an increase in proteobacteria and a decrease in Firmicutes and Bacteroidetes. We also noted a bloom of verruco microbia in C. difficile positive, and particularly C. difficile and norovirus positive cases. Of note, the presence of C. difficile was not associated with adverse clinical outcome in terms of length of stay or mortality. We concluded that children with diarrhea regularly carried toxigenic and non-toxigenic strains of C. difficile. We saw an enhanced microbiotal diversity and a change in milieu in the presence of C. difficile without any apparent morbidity. We also noted a specific microbiotal signature of norovirus and C. difficile co-infection. These findings were contrary to those seen in adults with C. difficile disease, raising the possibility that C. difficile could be a bystander in diarrheal disease in children. I'd like to thank the NIHR for their funding during the post where I did this research. There were many people at Alder Hey Children's Hospital and the University of Liverpool who were very generous with their time and expertise. But in particular, I'd like to thank Professor Anatang Carroll, who gave me my first post in research and has supported my career ever since. Thank you very much.